Hello and welcome to the third mini lecture on the scientific procedure. My name is Martin Hughes and this week we're going to be talking about data, more specifically experimental design. So we're moving on to the next part of the scientific procedure. Last week we discussed hypotheses, and at the tail end of the video last week I discussed experimental design, data collection and data analysis. So data can be split into multiple levels. This week we'll deal with data and we'll talk specifically about experimental design and introduce you to some key concepts. So, a quick disclaimer, I don't want to overwhelm you with information in this video. We will introduce a lot of information that may be brand new to you. Please pause, rewind and rewatch. But there's a lot that we don't cover too. And these videos are intended as a starting point for high school students or early undergraduate students that are unfamiliar with conducting their own experiment. So we know there's lots of things that we're not going to cover in this video, and we'd advise you then to maybe use these concepts as signposts to go and conduct your own research, do some further reading. There's plenty of videos online, lots of great materials. So this is just a starting point, but hopefully important to you. Okay, so we've already considered a lot of things before we've even collected a single piece of data. In the last video, we turned our questions into hypotheses. And we also looked at some graphs and mentioned having a control group. So we introduced some concepts and some terms that may be unfamiliar. In this video, we'll go into more detail about this and particularly discuss experimental design. So what is experimental design? Experimental design is a blueprint of the procedure that enables the researcher to test their hypotheses by reaching valid conclusions about relationships between independent and dependent variables. It refers to the conceptual framework within which the experiment is conducted. Okay, independent and dependent variables, what do I mean by that? An independent variable is defined as the variable that is changed or controlled in a scientific experiment. Independent variables are the variables that the experimenter changes to test their dependent variable. A change in the independent variable can directly cause a change in the dependent variable. Okay, this is important. I'll explain what they are using our simple explanations or simple examples in the last video. But before we go and collect a bunch of data, we need to make sure that we collect meaningful data. And this means you have to consider the design of your experiment carefully. And there's lots of ways to design an experiment. And we'll use our simple hypotheses from the last video to illustrate a few and introduce some key terms you should be aware of, including independent and dependent variables. So last week we talked about a Red Bull or a Coke or a sugary drink before bed and how that may disrupt your sleeping pattern. So let's just remind ourselves of those hypotheses. So our null hypothesis, or H0, was sugary drinks do not affect the sleeping pattern. Our alternative hypothesis, or H1, was that sugary drinks do affect the sleeping pattern. So in terms of independent and dependent variables, in this case, the independent variable, what we control, is the sugary drink. And the dependent variable, what we're measuring, is the sleeping pattern. So, we know what we're changing, the independent variable, and we know what we're measuring, the dependent variable. But how would we design this experiment? Well, we do have a couple of options. For example, we could use an experimental group and a control group. I think that's what I mentioned last week as well. So the experimental group, we'd have one independent variable, which is our sugary drink, and maybe 10 participants. And they'd be given the drink, and we'd monitor the sleeping pattern. Conversely to that, we'd also have a second group, we'd call the control group. They're not given anything before bed, no sugary drinks, and there'd be 10 participants there, and we'd monitor their sleeping pattern. So, an experimental group is the group in an experiment that receives the variable being tested, and one variable is tested at a time. The experimental group is compared to a control group, which does not receive the test variable. In this way, experimental groups are used to find answers in an experiment. So that's maybe quite intuitive. We'd give somebody, or the group, the thing that we're trying to measure, in this case, sugary drink on sleep pattern, and we'd have a control group that is not given the sugary drink, and we'd measure sleep pattern. So that's quite intuitive, and that would probably be quite a straightforward way to approach this experiment. But you remember from last video that we discussed other independent variables that may be affecting the sleep pattern, such as age, height, weight. All these things can affect the sleep pattern, the dependent variable. So if that was the case, if there was a lot of variables that we maybe wanted to control, a lot of other independent variables other than the sugary drink that could affect our dependent variable sleeping pattern, we may want to use what we call a matched pair experimental design instead. This is where each condition uses different but similar participants. An effort is made to match the participants in each condition in terms of any important characteristic which may be affecting the dependent variable. So gender, age, maybe not intelligence in this case, but weight certainly would as well. So we maybe use a design like this. So we'd still have our experimental and our control group. We'd still have the sugary drink in the experimental group, no sugary drink in the control group. We still have 10 participants, but those 10 participants are now the same age, the same gender, the same height, and the same weight. And that may be achievable, but it's probably extremely difficult to do in practice. Just think about all the different heights and weights you have within your own class or your own school. It may be quite difficult to match them up perfectly and get somebody who's the same height as me, the same weight as me, and the same age as me, all in the same experiment. So, realistically, the experiment and control group design is probably the most achievable. So our first experimental design. One way we can increase the credibility of our results, however, would be to repeat this experiment with different participants. In other words, replication. So we've got 10 participants at the moment. So if we had 10 people in the experimental group and 10 people in the control group, the total number of participants would be 20, and that's our sample size. And that's often notated as n equals 20. That's the total amount of people we have in our experiment. If we replicated this experiment 3 times, we would then have data on 30 people from the experimental group and 30 from the control group. And suddenly our sample size is now n equals 60. We've got 60 participants. And this would give us more confidence in our results because we've increased our sample size, which we'll discuss in a little bit on. But if we can increase our sample size, if we can replicate the experiment, it gives us more power. And we'll explain that in more detail. So, I've introduced some important terms, so we're just going to recap. The experimental group is the group exposed to a variable. The control group then is the counter to the experimental group, and they remain normal. Replicates are the repetition of the experimental condition, so the variability associated with the phenomenon can be estimated. So we want to be able to replicate our experiments, and I mentioned that very early on in the first video. Replication. Sample size. What is sample size? Sample size is the number of observations made in a sample. Sample size is an important feature of any empirical study into which the goal is to make inferences about a population from a sample. In practice, the sample size used in a study is determined based on the expense of data collection and the need to have sufficient statistical power. So basically what we're saying there in that last sentence is it'd be great if we could have lots and lots and lots, a large sample size, lots and lots of participants, but we have to kind of balance that with how easy that is to do. The cost involved in doing that, and we need to balance that up against our statistical power. So how many people, what is the minimum amount of people we need to be able to have accurate or appropriate statistical power? And we'll talk about that later on as well. But as a rule of thumb, the bigger your sample size is, the better it is for us when we come to data analysis. Okay, if we have, for example, one person in the control group drinking a sugary drink and one person in the control group, the experimental group drinking the sugary drink and one person in the control group, that one person in the experimental group may drink the Red Bull and have the best sleep of their life, and the person who didn't have anything may have a disrupted sleep. So are we actually having a fair comparison? Or if we increase that to 10, 20, 30 people, then these outliers would be brought in by the mean score of the rest of the group. So if we increase the sample size, we're less likely to pick up on these chance random anomalies, or we do pick up on them, they're not going to affect our overall result. So that's kind of what we mean, but I'll go into that in more detail. Like I say, there's a lot of information here. So if you want to pause, take notes, rewind, that's great. Hopefully you came across some of these terms before. If you haven't, there's lots of information on them elsewhere online, and we'll explain some of them in more detail as we continue forward. Okay, so example 2, back to our bamboo trees and pandas. So we talked briefly about that last week, but how would this actually look in practice when we are discussing experimental design? So this is now getting a little bit more complicated than our sugary drink example. So basically, what do we want to do? Let's remind ourselves. We want to examine the relationship between the number of pandas and the number of bamboo trees. So let's consider our hypotheses again. Our null hypotheses are H0. There is no relationship between the number of pandas and the number of bamboo trees. Our alternative hypotheses are H1. There is a relationship between the number of pandas and the number of bamboo trees. Okay. So, our independent variable is the number of bamboo trees. Our dependent variable is the number of pandas. So what we are measuring is the number of pandas and if the number of bamboo trees has an effect on this number. So that's our independent and our dependent variables. Okay, so we want to investigate if a relationship exists. But first we have to consider the size of the population. So we're talking about sample size again. How many pandas would be enough? So in 2015, the estimated number of giant pandas in the wild was 1,800. So is it realistic that we're going to survey every single panda? Probably not. We're not going to be able to get 1,800 pandas, but that's okay. What we're going to do is take a subsample of the population. And it may not always be possible. It's great that we've got that 1,800 figure, but a lot of animal species, we don't actually know the size of the overall population. So what's important here though is we investigate enough individuals that are representative of the population. So the individuals we're looking at, the amount of individuals we're looking at is basically a representation of the population as a whole. So for this question we may consider, well, 10% could be good. That's 180 pandas. 5%, that's 90 individuals. Is it 10% or 5%? Well, there's actually no correct answer here. However, it comes back to your critical thinking skills. And remember, we did our research before we designed our hypotheses. Well, during that process of doing your own research, hopefully you've came across other papers similar to the study that you're going to conduct. And it's important to consult this previous research and ask how many individuals did they use in their published research? How many individuals have been used in previous studies? And use that as a good ballpark figure for your own investigation. We already know we're not going to be able to sample all of them, but how many is a good enough amount for our sample size? And again, I'll reiterate, the larger your sample size, the more confidence you will have in your results, and it increases your statistical power. And we'll hammer that point again later on. But basically, if we know there's a minimum amount of pandas that we need to survey in this experiment, and we can go beyond that, fantastic. But we need to have a large enough sample size that allows us to do statistical analysis. Okay, so quite different from the sleep experiment. Okay, in the real world, this is going to be quite a complicated study, and you need to measure a lot of other variables that may be affecting panda distribution in an attempt to control the effects of these variables. So unlike the sleep experiment, there's going to be lots of other independent variables other than the number of bamboo trees affecting the number of pandas. Could be the season, could be temperature, rainfall, all these things, deforestation, all these things could be affecting the number of pandas in the area as well as the number of bamboo trees. So that's something we need to consider. But for the sake of simplicity, we're just going to look at this single independent variable. But this is something you need to be aware of when you're conducting your own experiment. Think about all the other things that may be affecting your dependent variable. What is it you want to measure, and what could it be affecting that? And if you can identify those things and control them, fantastic. It gives us more confidence that what your independent variable, the thing you're measuring, is having an effect because you've controlled all the other variables. Okay, so I have this little video here. So we know Sichuan, when they make a lot of great sauce. And Sichuan is the region that's home to the giant pandas. And you can see it's in southwestern China here. And you can see it's an absolutely massive region. So again, we've talked about sample size and how big our sample size should be. Now we're talking about sample site. So is it plausible that we're going to sample the whole of Sichuan? Probably not. So what we need to do is start considering sample sites. So we know within Sichuan, the Baifeng Valley is home to a lot of pandas. Okay, so now we are in Sichuan, but now we're going to become a little bit more accurate about where we're talking about. So we've limited the area to Baifeng Valley. But even Baifeng Valley, we look here, it's a massive expansive forest, a lot of topography. So where are we actually going to be doing our sample site? You can see a big hill here. So we probably want to be in this part of the valley. So again, we're going to just look at a specific area of the Baifeng Valley. So we may map out a region such as this size here, but again, are we going to be able to sample this whole area? Probably not. So what we might want to do is split this one cross section up into multiple sections, multiple sampling sites. We want to number these sample sites. So we've got 10 sample sites within the Baifeng Valley. Okay, that's fair enough. So we've got our sample sites. We maybe have a rough idea, hopefully, about the sample size. Now, how are we actually going to collect this data? And what is our data going to look like? So this is all stuff we need to consider before we set off into China to look at the pandas. Okay, so we've got our 10 sample sites. What's going to happen? Well, play it through. We're probably going to go to the Baifeng Valley and collect our data. You're going to record the number of bamboo trees and pandas in each of the sampling sites. So we've got 10 sampling sites. And this will probably take days of trekking through the forest, probably weeks, if not months. So you're probably going to want to recruit some help. You'll have a research team and all the rest of it. So all this stuff is going to cost money. You're going to have to put away time. You're going to have the right experts involved. So these are all things that we consider. But say we've done all that and we have a good idea about how we're going to do it, we can have a look at our data. So what would our data potentially look like? So we go to site 1. We count the number of bamboo trees, 76, and the number of pandas within that area, within site 1. We've got 7 pandas. So we go through to the 10th site, and our data may look something like that. Okay, so we graph that data, and it looks like we have a sort of positive relationship. But when it comes to statistical analysis, it shows us that there's actually no correlation based on what we found. Okay, we don't maybe have enough data points. So we say there's no correlation. As bamboo increases, there's no effect on the number of pandas. So we would fail to reject our null hypotheses. But in our heart of hearts, we know, oh, maybe there is something going on here. It kind of looks like as we increase the number of bamboo trees, the number of pandas does actually increase. So what we may do is revisit our experimental design and consider, well, can we add 20 more sample sites? Can we increase the amount of sample sites that we can analyse for this experiment? And that's going to be down to finances, and this is all things you need to consider beforehand. But say we do have the money and we can add 20 more sample sites. Suddenly we have a really strong, clear indication, positive relationship, positive correlation. Yes, as bamboo increases, so does the number of pandas. So we can reject our null hypothesis that there is no relationship. So this comes back to sample size again. And this is why it's so important to figure out what it is or how many samples you're going to be able to collect. So if you only have a few data points, it's going to be hard to observe any pattern. And I think I reiterated this last week as well. It's why we never accept our null hypothesis. You have just failed to reject it based on your own data. It doesn't mean something isn't happening. You just may not have picked up on that in your experiment. So we can test these patterns. So I know I've just used graphs and said we accept or reject. So we accept or reject, we fail to reject or reject based on statistical analysis. And the more data we have, the more statistical power that gives us. And we'll explain that in a later video when we collect our own data. But it's something to be aware of. Something I haven't mentioned here is types of data. And this may be something you've came across before. In the panda experiment, the data collected on the pandas and the bamboo trees was what we called count data. Count data is sometimes referred to as discrete data. It can be considered numeric, and it must be a distinct number. Okay, it can't be negative. For example, you can't have 1.5 pandas or -1 pandas or 1.5 bamboo trees. It's either 1 or it's 2, and you can't have a negative amount. It'd either be 0. It'd have to be 0. Continuous data, on the other hand, is also numeric, but it can take any form, including negative integers. Like temperatures, we may have 14.6 degrees Celsius or -13.8 degrees Celsius. We have multiple numbers after the decimal point, significant figures. Height as well could be continuous data, and weight. Okay, so you can hopefully see the difference there between count and continuous. Excuse me. And categorical data is when data falls into a category. Like eye colour, for example, you'd either have blue, green, brown or hazel eyes. Gender, you're either male or female. Age, you're either a child, a teen, a young adult. So we have distinct categories. So we call that categorical data. And the last one you maybe not have heard of or you might not come across is binary data. And this is where we only have 2 categories, a 1 or a 0, yes or a no, alive or dead, infected or uninfected. Okay, there's only 2 states that you can be, and we call that binary data. So before I move on, again, a lot of information here. Please take your time with this, rewatch the videos. I'm not trying to overwhelm you, but these are really important concepts and terms that you need to get to grips with if you want to go out and conduct some really good research. Hopefully some of this is familiar, and if it's not, it will be. Although our dependent and independent variables in our panda example were both examples of count data, you could have a combination of the two. Okay, you don't have to have count data for your independent and dependent variable. You could have categorical and count, categorical and continuous, and it's important to consider what type of data you have prior to collecting it. So if you know it's going to be count or continuous, and you know you're going to have a few categories, put all that down in writing and maybe run through some hypothetical scenarios, and we call that using dummy data. So like I've done with the panda experiment, I've made that data up, but it gives me a good indication of maybe what I'd see in the real world, and I can start messing around with that. So when I do collect real data, I know that it's going to work. I know how I'm going to analyse it. And the analysis of data is really critical, and we'll talk about that next week. In the next video, we're actually going to put all of this together. Okay, everything we've learned so far is going to go into our bird feeder example that we mentioned in video 1. And you guys are actually going to be able to extract your own data from the videos that we're going to upload after that. So hopefully, after next week, we're now starting to get you guys involved. You'll be able to analyse your own data and collect your own data, and then we can talk about analysis. And then finally, we can talk about, well, how do we write this up? How do we illustrate the information that we've had through a report, which a lot of people are put off by as well. So we'll cover all that in the next video. So thank you again for paying attention. If you could like, subscribe and follow us, that'd be awesome. Maybe leave us a comment. You can follow us on Twitter at inspiring_eco and myself at Dr Martin Hughes. Thanks very much.